Okay. Okay, so to investigate the effects of various cooking procedures on the nutrients and phytochemical compounds in red cabbage. Okay, so if we were to look at the experimental flow chart. Okay, so you have the preparation of red cabbage and then the cooking treatments consisting of fresh cut, boiling, stir frying, microwaving, steaming, and then the materials, and then the storage, and then frozen material storage, frozen by liquid nitrogen, and then kept in polyethylene bag at 20 degrees C. Okay. Okay. So, okay, the methods of analysis. Okay, you can see this. Okay, and then factor. Okay, cooking method. So only one factor. Number of treatment. Okay, is 5. And then treatment level, fresh cut, boiling, steaming, microwaving, and stir frying. Okay. So how many factors? One factor. Okay, it is a cooking method. Okay, so it is the cooking method. Okay, what about the material storage? Okay, so how many methods of storage? Only one. Okay, so frozen by liquid nitrogen, and then after it has been frozen, then it is being kept at in a polyethylene bag at -20 degrees C. Okay, so it is only one. Okay, one factor. Okay, how many levels or types of the cooking method? They have 5. Okay, so 5 level or type. Okay, so meaning that number of treatment is 5. Okay, so number of replication is 3. So the number of experiment, okay, that you have to do is 5 times 3. So 15 experiments that you have to do. Okay, how many responses? Okay, responses are those that you measure. Okay, so you have color, total anthocyanin content, TPC, DPPH, vitamin C, TSS, and then reducing sugar content. Okay, and then total glucosinolates. Okay, so you have 1, 2, 3, 4, 5, 6, 7, or 8. Okay, I'm not sure whether this is going to be considered as one or two separate ones. Okay, so if it is two separate ones, then it is 8 responses. Okay. Okay, so let's take a look at the quiz. Okay, so will a two-way ANOVA be suitable for this study? Okay, the answer is no. Okay, why? Because it is a one-factor experiment. Okay, because it is a one-factor experiment. Okay. Okay, any questions? Is everybody clear? Okay, so next presenter is Kamarina. Okay, Kamarina, please. Is Kamarina here? Okay.
Can you see my screen?
Yes.
Okay. Assalamualaikum dan selamat pagi. Nama saya Nur Kamarina binti Abdullah. Nama matrik saya adalah P115092 dan hari ini saya akan membentangkan kajian saya yang bertajuk potensi selulosa bag. Oh, sorry. Sekejap. Sekejap. Okay, potensi selulosa bag. Saya buat macam ni je boleh tak? Sebab dia macam auto. Okey ke kalau saya macam ni je? Hello?
Maksud you tak boleh tu ke?
Dia auto.
Auto maksudnya? Slideshow tu dia boleh klik. Cuba klik tengok.
Ni. Dia auto gerak sendiri.
Okay, so macam ni? Okay.
Ni saya tak tekan, dia gerak sendiri.
Okay, you ada letak setting tu kat situ tu. Okay, boleh? Masing-masing pun cuma besarkan je. So masing-masing, each of you has their own copy. So we can proceed.
Tajuk saya ialah potensi selulosa bakteria daripada hidrolisit tandan kosong kelapa sawit yang ditambah dengan ejen antimikrobial untuk memajangkan jangka hayat makanan. Jadi bagi kenyataan masalah, pembungkus adalah alat utama yang dapat memanjangkan jangka hayat makanan kerana ia bertindak sebagai penghalang fizikal daripada oksigen, lembapan, sebatian kimia
Sepatutnya Gamarina dalam English
sebab ni kajian saya terus ambil dalam proposal saya, sebab kalau nak translate, dia macam banyak perkataan yang saya macam tak sure. Walaupun saya dah guna ikhlas dengan...
Kalau tak sure, that is your responsibility to make it sure.
Alright, okay.
Okay, proceed.
Dan mikroorganisma yang merosakkan makanan. Sebatian antimikrob semula jadi dan polimer terbiodegradasi menjadi trend terkini dalam bidang pembungkusan antimikrob kerana dapat kerisauan pengguna yang berkaitan dengan kesihatan jika ejen antimikrob komersial dan polimer sintetik digunakan sebagai bahan pembungkus. Jadi menurut Ikin dan Esma, penambahan ejen antimikrob sintetik dalam makanan berkesan untuk menghalang pertumbuhan pelbagai mikroorganisma, tetapi pengguna memerlukan produk makanan yang diproses secara minimum, bebas pengawet dan tahan pada jangka masa yang lama. Pembungkusan aditif adalah pendekatan inovatif untuk mengekalkan dan memanjangkan jangka hayat produk. Pembungkusan makanan sedia wujud dalam masyarakat kini telah menyebabkan penghasilan sesampah yang besar kerana ia tidak terbiodegradasi dan ini dapat dibendung dengan penghasilan polimer terbiodegradasi daripada TKKS iaitu tanah kosong kelapa sawit untuk menghasilkan selulosa bakteria. Dan atas sebab inilah kajian ini harus dilakukan. Jadi bagi objektif saya ada 3. Pertama ialah menghasilkan hidrolisit daripada TKKS secara sakarifikasi berenzim untuk pertumbuhan Comagete bacteroxylinus. Kedua, menguji keberkesanan ejen antimikrob semula jadi dalam menghalang pertumbuhan bakteria dan kulat. Yang ketiga ialah mengkaji keberkesanan penggunaan selulosa bakteria yang ditambah dalam ejen antimikrob dalam memanjangkan jangka hayat makanan. Seterusnya ialah carta alih ataupun flowchart. Pertama sekali, selepas kajian perpustakaan dengan penyerahan cadangan penyelidikan, penyediaan sampel TKKS akan dilakukan di mana TKKS ini akan dicari-cari untuk mengurangkan saiz dan dikeringkan. Kemudian akan dikisar. Seterusnya TKKS akan dikenakan perawatan menggunakan natrium hidroksida pada kepekatan 5% dan akan perawatan akan dijalankan pada suhu 121 degree Celsius, 117 kilopascal menggunakan autoclave. Kemudian ialah sakarifikasi berenzim menggunakan enzim accelerase 1500. Kemudian ialah kita akan terus ke ujian gula penurun menggunakan kaedah asid dinitrosalicylate iaitu DNS. Kemudian ialah penapaian K-xylinus. Ia akan ditapai selama 8 hari dan akan penapaian akan dilakukan selama hari yang pertama, hari kedua, hari keempat, hari ke-6 dan hari ke-8. Dan kita akan uji dia punya penentuan pH dan gula. Kemudian adalah penghasilan ekstrak ejen antimikrob. Kita akan menggunakan daun kesum dan daun serai dan kita akan gunakan juga satu ejen antimikrob komersial iaitu potassium sorbate. Dan kemudian kita akan rendamkan selulosa bakteria dalam ejen antimikrob dan kita akan lakukan kajian penyimpanan roti menggunakan ejen antimikrob yang menunjukkan inhibition zone yang tertinggi dan analisis data akan dilakukan. Bagi ujian statistik akan menggunakan Minitab versi 20 dan saya akan menggunakan analisis varian sehala dan dua hala iaitu ANOVA, dan ujian Turki akan digunakan untuk menentukan perbezaan ketara antara rawatan pada tahap keyakinan 95%. Bagi DOE, bagi ejen antimikrob aktiviti dia, saya akan gunakan completely randomized factorial design dan faktor dia adalah ejen antimikrob iaitu daun kesum, ekstrak daun kesum, daun serai dan potassium sorbate bersama dengan bakteria dan atau kulat, iaitu saya akan gunakan E. coli, Salmonella dan juga Rhizopus kulat. Dan saya akan lakukan sebanyak 3 replikasi dan ada 27 sampel. Seterusnya ialah rujukan dan itu saja bagi presentation saya.
Okay, you punya slide ni saya pun tak boleh nak baca sebenarnya, handout yang you bagi ni. You just copy and paste ni kan. Yang you ada, you punya carta alih lah. Okay, so saya tak rasa it is suitable. Okay, I don't think the other students also can read what you are, your handout. Okay, so we will skip this for the time. Okay, what I want you to do is to prepare another handout so that it is readable in English and email it to me. And then I will email it to the other students for along with the quiz questions. Okay, Kamarina?
Okay, Prof.
Okay, so we'll go straight to the next presenter. Okay, Mai Saidatul.
Boleh nampak ke slide saya? Hello?
Yeah. Okay, boleh.
Okay. Assalamualaikum and good morning everyone. My name is Masadatul and today I will present the DOE for my research. My research title is Effect of Organic Solvent on Antioxidant Content and Composition of Essential Oil in Different Parts of Serai Aceh, Elytropsis smithy. E. smithy is a zingiberacea herbaceous plant that is easy to grow. However, the public is still unaware about the existence of E. smithy as a herbaceous plant. The research conducted on E. smithy also limited. Therefore, information and data on the content and benefit of E. smithy as herbaceous plant are limited. This research has 3 objectives. First is to determine the optimal organic solvent to extract E. smithy for determination of antioxidant content. Second, to determine the antioxidant content for different parts of E. smithy. And third is to determine the composition of essential oil for different parts of E. smithy. This is the experimental flowchart for this research. Basically, there will be 2 experiments conducted. So for first experiment, the root, leaf, and stem of E. smithy will be extracted by using 3 different organic solvents, which is methanol, ethanol, and dichloromethane. Then the extract will be determined for its antioxidant content by using DPPH method, FRAC method, and ABTS method. The extraction also will be determined for its flavonoid content and phenolic content. For second experiment, the root, leaf, and stem of E. smithy will be hydrolyzed to get the essential oil. Then the essential oil will be analyzed by using GCMS. Next is statistical test. The data will be analyzed at 95% confidence level by using Minitab software version 18. The data will be analyzed by using two-way ANOVA, and Turkey test will be used to identify the difference. The correlation coefficient between antioxidant content and total phenolic content, as well as the total flavonoid content, also will be determined. Next is design of experiment. For the first experiment, there will be 2 factors involved, which is type of organic solvent and also part of E. smithy. The response are antioxidant content, flavonoid content, and phenolic content. The number of replication is R3, so the total sample will be 27 sample. For this experiment, the DOE that will be used is split plot design with a type of organic solvent as the whole plot and part of E. smithy as the split plot. For the second experiment, there is only 1 factor involved, which is part of E. smithy. The response are composition of essential oil of different part of E. smithy. The number of replication are 3, so the total sample will be 9. And the DOE that will be used is completely randomized block design. Okay, thank you.
Okay, thank you. Am I- Okay. So for this, mine showed two design of experiments for two aspects of study. But for this quiz, we'll just concentrate on the first one, extraction and antioxidant activity of yeast media. Okay. Okay. So any questions regarding the first part? The first part only. Okay. Before we start our quiz.
I have a question. Prof, can I ask a question?
Yeah, sure. Go ahead.
I would like to ask Mai what does she mean by the split plot design? Can she explain a bit?
Okay, hello. So split plot design mean that like me, I got two factor, which is one factor it was quite difficult to manipulate. So if I do the split plot design, so I can decrease the number of factor that we need to manipulate. For example, instead of I kalau I use the factorial design, so I can I will macam dia macam random kan. Tapi kalau I guna split plot design ni, maksudnya okay, kalau factorial design tu dia macam okay, kita akan buat untuk first replication. So untuk first replication tu kita akan randomizekan dia punya treatment. Replication je kita fix. So kita kena randomizekan methanol, ethanol semua extract 3 different sample. Tapi kalau saya guna split plot design ni, so maksudnya untuk yang replication pertama tu run order dia macam kita still randomizekan dia punya yang sample tu, tapi untuk solvent tu kita macam kita boleh fixkan. Maksudnya kita boleh, maksudnya kalau untuk eksperimen pertama tu methanol saja, tapi methanol tu kita guna different part lah, macam akar, daun, batang macam tu. Lepas tu baru ethanol. Macam tu, dia tak macam kalau factorial tu kita macam maybe ethanol dulu, lepas tu methanol dulu, lepas tu ada chloromethin. Dia macam tukar-tukar. Dia macam susah lah nak manipulate sikit.
Okay, so maksudnya guna fix solvent, lepas tu fix replication. Maksudnya saya contohnya guna methanol dulu, lepas tu buat first replication untuk semua parts. Macam tu ke? Lepas dah habis baru pergi ke ethanol, buat first replication, buat untuk semua parts. And then after that, macam tu ke?
Dia maksudnya macam kita randomizekan dia punya sample tu. Tapi sebab kita dah overplot dia, kita dah setkan sebagai solvent. So solvent tu antara 3 tu lah, sama ada ethanol ke methanol ke.
Maksudnya yang random tu cuma part of the plant, right? Sama ada root atau stem, yang tu yang random kan?
Tapi dia still ada run order lah. Run order dia. Dia still kena buat lah. Okay.
Okay. Any other questions?
Sorry. Siapa tadi? Amai. Apa so maksudnya replication pun randomize jugalah kan untuk yang apa ni split plot ni kan? Betul tak?
Untuk yang split plot ni dia macam factorial design je. Maksudnya kita akan buat untuk replication 1 dulu.
Oh, buat replication 1 juga dulu lah. Maksudnya ethanol, lepas tu replication 1 dulu, root, lepas tu stem, macam tu lah.
Dia macam A3 tu, tapi tiga-tiga tu untuk first rep punya. Tapi untuk 3 different sample tu lah. Tapi semua first.
Tapi maknanya ethanol first rep, lepas tu semua root, semua parts. Lepas tu ethanol second rep, semua parts.
Itu untuk yang lain. Betul.
Okay.
Untuk yang lain lah.
Alright, thank you.
Okay, any other questions? Okay. So if everybody's clear, then I'll show you the. Okay. Okay, so you have 10 minutes. Okay, time is up. So please submit your answers. Okay, so everybody has submitted. Okay, so let's discuss this. Okay. Okay, so this is my research. Okay, effect of organic solvents on antioxidant content and composition of essential oil in different parts of Serai Aceh. Elateriopsis smithiae. Okay. Okay. She has 3 objectives. Okay. And over here, she's showing 2 parts of the experiment. Okay, so we'll just concentrate on the first part. Okay, so for this one, okay, so extraction and antioxidant activity of yeast methiae. So she has 2 factors. The first is type of organic solvent, that is methanol, ethanol, and dichloromethane. And then the second factor is parts of yeast methiae, that is the roots, leaves, and also stems. Okay, so 2 factors. What are the levels of the factors? Okay, so for the first one, you have 3 types. Okay, 3 types. Okay, and then for the second factor, that is the parts, also have 3 types. Okay, so for this particular study, all the data, all the levels are non-continuous. Okay, non-continuous. So these are the 3 types of organic solvents and 3 types of parts of the plant. Okay, and then the response, she has 3 responses. Okay, antioxidant content, actually 5 responses. So antioxidant content for DPPH, based on the DPPH, ABTS, and also FRAP, and also flavonoid content, and also total phenolic content. Okay, so how many treatments? Okay, so each factor has 3 types. So how many treatments? She has 3 times 3. That is 9 treatments. Okay, number of replications, 3 replications. And then, so 9 times 3 equals to 27 samples. Okay. Okay, so the DOE is split plot design. She's where she's taking the whole plot as the for organic solvent as the whole plot, and the parts as a split plot. Okay. Okay, we'll discuss that later for the split plot based on the questions by some of the students. Okay, but let's look at the quiz first. Okay, so if one of the factors is omitted, what type of statistical tests would you use to compare between means? Okay, so if we were to look at this, if you were to omit one of the factors, for example, if you were to omit the parts, so now we only have one factor. Okay, one factor with three types. Okay, so with 3 types. So the question is, what type of statistical test would you use to compare between means? Okay, so how many sets of data are we comparing? Okay, so it has how many. If I omit the parts, then only types of organic solvents is left. Okay, so what are the treatments? Treatments are 3 treatments, that is methanol, ethanol, and dichloromethane. So how many sets of data are we comparing? We are comparing between 3 sets of data because we have 3 treatments. Okay, so we are comparing between methanol and ethanol and dichloromethane. So for each methanol is 1 set of data. Okay, because you will be doing 3 replications. Okay, and then this is 1 set of data compared to comparing it to ethanol, another set of data, and also comparing it to dichloromethane, another set of data. So because we are comparing between more than 2 sets of data, so t-test is not suitable. Okay, so t-test is not suitable. So what do? We should do an ANOVA, analysis of variance. So because it is a one factor, okay, experiment, so should be a one-way ANOVA only. Okay, cannot do for two-way ANOVA. Okay, similar to what to the quiz question for Suyen. Okay, so one-way ANOVA. Okay, so one-way ANOVA, and then can ANOVA only shows whether there exists a significant difference among the means or not. Okay, but it cannot compare between different means. So this has to be followed by a multiple means comparison test or a post hoc test. Okay, whether it is a Duncan, Tukey, Fisher, LSD, or other types of post hoc or MMCT tests. Okay, okay. So that should be the answer. Okay, so the answer should be, okay, what type of statistical test should be ANOVA followed by an MMCT. Okay, why? Because you are comparing between more than 2 sets of data. Okay, any questions? Okay, so regarding the split plot. Okay, so regarding the split plot, okay, I'll just make I'll show you some a basic, not a basic, just a preliminary way of doing it. Okay, so this is a split plot. Okay, why? Because based on the practicality of it, it is difficult for Mai to do a factorial design. Okay, so if it is a factorial design, meaning that she has 27 samples. Okay, if it is a completely randomized factorial design, then she has to play around with 27 samples, and that is too much. It's quite difficult. Or if she were to do a factorial design using blocks, taking the replication as blocks, so meaning that she has to manipulate 3 times 3, okay, because 3 types of organic solvent times 3 parts, okay, so 9 treatments, okay. So, okay, maybe the manipulation of organic solvents is difficult, okay, so that is based on what Mai has told us, okay. So if that is the case, then she can use split plot, okay. So for split plot, remember the randomization is in sequence. Randomize the whole plot first and then randomize the split plot, okay. So which one do you do for split plot, which one is the whole plot and which one is a split plot? You don't need to randomize, okay. You can choose, okay, and usually we will choose the factor that is the most difficult to manipulate as the whole plot, okay. So for this one, for Mai, it is the organic solvent that is the most difficult to manipulate, okay. So she designate the whole plot as the organic solvent, and the split plot is the part of the plant, okay. So when she goes into the lab, okay, and prepares the run order, so what will happen is that, okay, so if this is the run order, okay, so you have 1, okay, and then you have, okay, so you have all the rest up to 27, okay. So you have 27 experiments, right? Okay. So, and then for this split plot design, she wants to do it according to block, because she only wants to handle 1 replication at a time. So take the replication as the block, okay? So you will have, this is the whole plot, okay? Designate the whole plot, okay? Okay, so I'll just put it up there. So the whole plot is solvent type. Okay. Can all of you see? Is it big enough? Okay. What happened? Oh, I cannot increase it. Okay, so maybe I'll just make it larger. Okay. Is that more readable?
Yes, bro.
Okay. So this is the run order. Okay. And then you have the whole plot. Let me just, okay, write text. Just do this and also do this. Okay, so that is the whole plot, and then you have your split plot. Split plot. Okay, that is the parts. Okay, the plant parts. Okay, so okay. Sorry. Okay, so for the whole plot, okay, so the solvent type. So which solvent do you do first? Then you have to randomize. Okay, then you have to randomize. Okay, so first randomize the solvent, and then within the solvent, randomize the split plot. Okay, so for example, she did a randomization and it comes out that the first solvent, okay, randomly is ethanol. Okay, so for ethanol, then this is the whole plot. So meaning that she has to do, oh, sorry, she has to do all the ethanols first. Okay, so this is the whole plot. But within this whole plot, she has a split plot. Okay, that is the part. So among the ethanol, okay, so which part does she do first? Then she has to randomize. Okay, roots, leaves, or stems. So randomize it. For example, she got she picked straws, and then she got stems. Okay, stem, and then pick another straw, she get the roots. Roots, and then after that the leaves. Okay, the leaves. Okay, so then finish for ethanol. Okay, and then after ethanol, okay, then she randomized again, and then when she picks straws, then it comes out as methanol. Okay, so then she do all the methanols. Okay, but okay, for the split plot, she should not copy this one. Do not copy this. You have to randomize it again. Okay, so when you randomize, for example, you get like roots, okay, and then leaves, okay, and then of course it has to be stem. Okay, and then the last one will be, oh, sorry, replication. I forgot. Okay, so this is the replication. Okay, so all of this will be replication 1 because she's doing it in a block. Okay, taking the replication as block. Okay, so and then this is the dichloromethane. Okay, DCM. Okay, so do for all the dichloromethanes, but over here you have to randomize again. So for example, you got the stem, and then the leaves, and then the roots, for example. Okay, and then after that, so finish your first replication. Okay, so meaning that for ethanol, maybe she has to do it under the fume hood or etc. I'm not sure. Okay, but manipulation of the solvent will be easier because you only have to handle ethanol once, and you do it for all the different parts. Okay, and then you handle methanol, and then you do for all the different parts. If it is a factorial design, then for this first one, maybe it is ethanol stem, and then comes DCM stem, for example, and then comes methanol leaves, for example. Okay, so that is factorial design. Okay, because even though you do factorial design using blocks, but still all the nine different treatments has to be randomized. Okay, so meaning that what she will be doing in the lab is that the first run order is using ethanol, and then she has to switch to methanol, and then maybe back to ethanol, and then DCM, etc. As opposed to doing a split plot, she only handle the ethanol, okay, and then the methanol, do it for all, and then the DCM, do it for all. Okay, and then do it for the second reps. Okay, 9 plus 9, 18. Okay, for the second rep. Okay, but do not follow this sequence. Okay, you have to pick straws. Okay, for example, you pick one and then you get like methanol. Okay, then it will be methanol. Pick another straw. Okay, and then it comes out DCM. Then you do for DCM. So all of these are your whole plot. And then of course, lastly will be for the ethanol. Okay, but within this whole plot, then to decide the split plot, you have to randomize again. Okay, so for example, comes out for leaves, and then for stems, and then for roots, of course. Okay, and then this one for roots. Okay, and then for stem, and then for leaves, like that. Okay, so randomize everything. Okay. Okay, so that is just an example. Okay, any
Sorry,
questions?
Doctor, can I ask? Yes, I have a question,
Yeah.
Prof.
Mhm.
Okay, I understand the practicality of this. I get it, the whole plot. But okay, just now you said the solvent type maybe it's difficult for Mai to handle. For example, the ethanol, she only wants to do it once. Then why can't she do the first rep, second rep, and third rep with all ethanol, and she just randomize the plot, yeah, the plant parts. Can't she do that?
Okay. No, she cannot, because when if she were to do it like that, then there is a concern of bias, because all the ethanols will be done first, and then followed by the methanols. Okay.
Yeah, I get it.
If
But then
uh,
the reason of doing that is because we want to handle that one particular solvent at one time only. Cannot, is it,
Yes,
Prof?
yes, of course. Yeah, we want to do it, but not at the expense of incorporating or increasing the possibility of bias.
I see.
We
Okay.
can reduce the we can simplify the way that we do experiments, but within the constraints of the design of experiment according to the statistical principles.
All right. Okay.
Okay. Okay. Any other questions? Okay. So if there's no question, then we'll proceed to the next presentation. Okay, the last one that is for today. Okay, that is Hajar. Okay.
Okay. Assalamualaikum warahmatullah. Can everybody see my slide and screen?
Yes, we can see it.
Okay. Thank you, Prof. Okay, so I shall start. Okay, so for today I will presenting a topic about from a journal, a topic about the effects of calcium chloride treatment on softening in red raspberry fruit during low temperature storage. Okay, so what are the problem statements? For the first one, red raspberry has a high nutritional value and has an anti-cancer effects. So it's easy to deteriorate and soften during storage, which can lead to a short shelf life. So from the previous studies, by lowering the temperature during storage, actually can extend its shelf life. And fruit softening also affects the degradation of cell wall and can affect the loss of cell structure. So what are the objectives for these studies? So the first one, to study the effects of calcium chloride treatment on softening of red raspberry fruit during storage at 4 degrees Celsius. For the second one, to study the role of calcium chloride treatment on cell wall degradation of red raspberry fruit during low temperature storage. So for the flow chart of this experiment, we start from harvesting the red raspberry fruit, which we distribute by the color, by the size, and free from any defects. Then second one, then next step we place the fruit in a PET storage boxes. Then we transport them to the laboratory, and we form into two groups of raspberry, which are for the first one 0.2 mole per liter of calcium chloride, and for the second one the control condition with the ionized water. So both of these factor are carried out for 3 minutes during storage at 4 degrees Celsius. Then after that, all the fruits actually stored in 4 degrees Celsius, which is this is the constant temperature. And after that, the samples take within 2 days. So, what are the treatments used in these studies? Firstly, to determine the firmness and respiration rate, and the second one to determine the physiological measurements. So for the replications used in the studies, three replications, and for the treatments, we times by two, and we got 18 samples. So the physiological measurements, actually we got three. So the first one is the content of protopectin, soluble pectin, and cellulose. And second one's the cell wall, which degrading enzyme on starch. And the third one, the content and AM activities. So for the design of experiment, the factors used for these studies are the calcium chloride treatment and the control treatment. Uh, for the treatment itself, we got two level of treatments, which are first one, the firmness and respiration rate, and second one is physiological measurements. So what is the design of experiment used in these studies? In these studies is completely randomized factorial design. So what the statistical tests involved in the studies? So we use t-tests, which we want to compare the differences between two groups for the control and the uncontrolled. So the software used for the studies is SPSS version 21.0. Right. So that's all from me. Thank you.
Okay, thank you, Hajar. Any questions from the students? Okay, so everybody is clear. Okay, so if that's the case, then we proceed with our quiz. Okay, so you have 10 minutes. Okay, so time is up. Okay, so please submit your answers. Okay. Shafika, have you submitted?
Yes, Prof.
By email or by WhatsApp?
Email, Prof.
Oh, okay. Okay, Sin. Okay, thank you. Okay, so quiz. Okay. Okay, so this is Shafika's project. Oh, this is from paper. Shafika, sorry, Hajar. Okay, so this is from a paper. Okay, so the objectives to study the effects of calcium chloride treatment on softening of red raspberry during storage. Then to study the role of calcium chloride treatment on cell wall degradation of red raspberry. Okay. Okay, so these are the flow charts. Okay, and then design of experiment, factors, two factors. Okay, so the factor first is calcium chloride treatment and control treatment. Okay, the calcium chloride is 0.2 mole per liter. Is it correct, Hajar?
Yes, Prof.
Okay, okay. Okay, so remember, if it is a factor, then it is not constant, right? Okay, if it is a factor, then it is not constant. Okay, so if we were to say that these are two factors. So the first factor is calcium chloride. Okay, how many levels are there for this factor? It's only one level, right? That is 0.2 mole per liter of calcium chloride. Okay, for the second one, control. How many levels of control?
Just one.
Only one level. Okay, so meaning that these are not factors. Okay, so these are not factors. Okay, but what is the factor? What is changing? Okay, the rest has to be constant. Okay, but what is changing? These are changing. Okay, so what are these? These are treatments. Okay, you can just give a general name. Okay, treatments. If it is very specific, for example, that you have different concentrations of calcium chloride, then you say it is a concentration of calcium chloride. But in this case, you only have two. Okay, one is calcium chloride, the other is no calcium chloride. So this you can make a general category and name it as treatment. So meaning that over here, you have two treatments. One is control, meaning that without treatment. The second is calcium chloride, meaning that you are adding or applying calcium chloride. Okay, so meaning that this is actually a one factor experiment. What is the factor? The factor is treatment. Okay, what are the levels? The levels or type is, one is without treatment or without calcium chloride, zero calcium chloride. The second one is 0.2 mole per liter calcium chloride. Okay. Another way for you to determine whether it is a factor or not, because remember, factors are things that are aspects that we want to investigate. In order for us to do so, then we need to vary the factor. That's why after factor, there is a level. Okay, after factor, there has to be a level or type. Okay, and then it is then based on whether it is a one factor experiment or a two factor experiment, then you will have a treatment. Right. Okay. So if we were to say that these are two factors, calcium chloride treatment is one factor, then what are the levels? There's none. Right. So meaning that it is not a factor, because factors always have levels or type. And the combination of levels or type, you will get the treatment. Okay. Okay. Similarly, if you were to say that this is the factor, then meaning that then you have to ask, what is the level? So there is none. Okay. So it is not a factor. Okay. But what you want to do is you want to designate the treatment as a factor. If the treatments are a factor, then what are the levels or type? The levels of type is first, control or zero calcium chloride. The second is 0.2 mole per liter calcium chloride. Now you have a proper design of experiment. Okay, because you want to compare between these two. So what are the treatments? Because it is a one factor experiment, so the levels or types are the treatments. So meaning that you have two treatments. Okay, bukan two treatment levels. So this one you put two treatment levels, but you didn't put what are they. Is it firmness? Okay, so meaning that you have one factor that is treatment, and then what are the levels? The levels should be zero calcium chloride, and the second one is 0.2 mole per liter calcium chloride. So what are the treatments? Okay, the treatments are the levels or type. Okay, so you have two treatments over here. So two treatment levels. Okay, so you say that firmness and respiration rate and physiological measures. Okay, remember, treatments are things, aspects that we impart on the samples. Okay, we apply on the samples. Temperature, heating, cooling, extraction, solvent, different solvents, all of this we impart on the samples. Okay, firmness and respiration rate. Do we impart it on the samples? So over here, to determine, when we say determine, meaning that we measure. How do we determine? By measuring. When we measure, once we measure, then it is not a treatment anymore. It is a response. Okay, so this is the response. The responses are things, aspects that we measure. So firmness, we measure firmness. We measure the respiration rate, and we measure the physiological measurements. Okay, so all of these are aspects that we measure. So these are not treatments, rather they are responses. Okay, so it is for this particular study, it should be a one factor experiment with two treatments, that is control and also calcium chloride, with 1 to 3 responses, that is firmness, respiration rate, and physiological measurements. Okay, so the design of experiment used should be a one factor design of experiment. So this is not correct. Okay, it cannot be a completely randomized factorial design. Okay. Okay, Hajar, clear?
Okay, Prof. Thank you.
Okay. Okay, so let's go to the quiz. Okay, so will a completely randomized block design be suitable for this study? Okay, so the answer is yes. Okay, why? Because it is a one factor experiment. Okay, so completely randomized block design is a one factor experiment, similar to CRD, completely randomized design. So we can use either one of these, okay, depending on the convenience and the limitations that you have in the lab, okay, for this particular study. Okay, so yes, why? Because it is a one factor experiment. Okay, any other questions?
Prof, sorry. So kita kena randomize the factor, kita kena randomize the factor kan? I mean the treatment kan? The response kita tak payah randomize pun tak apa kan?
Tak apa, yes.
Okay, alright, thank you.
Because design of experiment is how you apply your treatments. Okay, after that you can collect all of them as long as you have justification, scientific proof that when you store your samples, okay, after treatment, then in a condition that will not affect the data, the results. For example, if you are going to measure the antioxidant capacity, then you store them at a low temperature in a dark place. So it should be okay. Okay.
Thank you, Prof. Thank you.
Okay. Okay. Any other questions?
Prof, I have a question. So if this is
yes.
a one factor experiment, then just now you said it shouldn't be CRFD. It should be a block design, right? I mean, one factor design.
Yes.
Okay, so what's the difference between the one factor design and a block design?
I'm sorry?
Then what is the difference between the one factor design with a block design?
A one factor design is where you only have one factor. So
Okay.
a one factor design, you can use blocks in a one factor design. You can also use blocks in a more than one factor design. For example, if it is a one factor design, then you can use a completely randomized design, a CRD, or a completely randomized block design, or CRBD. So these two designs only cater for one factor designs. But if it is a more than one factor, but if you have more than one factor, so you can use a factorial design. A factorial design, you can do it either complete randomized factorial design or factorial design using blocks. So similar to split plot, like the one that we just discussed, you can do a split plot, but completely randomized, or you can also do a split plot using blocks, which is much more easier, of course. So blocks,
we just look at the factors, how many factors there are, right, to determine which design.
Yes, first and foremost, you have to look at the factors, because the design of experiments, they are divided into two. First is one factor experiment. The second is more than one factor experiment. Okay. So based on those, then within the one factor experiment, you have, okay, let me share over here. Am I still sharing? No. Okay. Okay. So, okay. Okay, so for example, okay, when we talk about design of experiment, you can divide it into, okay, one factor, DOE, and then more than one factor, DOE. Okay, so for one factor DOE, you have completely randomized design, you have a completely randomized block design. Okay, so more than one factor, you have many. Okay, for example, the most basic is a factorial design. Okay, for factorial design, you can do completely randomized, or you can do a block design. Okay, so similarly, you also have a split plot. You can also do it whether completely randomized or with blocks. Okay, and then you also have more special. These are more general, but you also have more specialized designs such as CCRD. You also have mixture design. Okay, you have Taguchi, you have box banker, and all of those. Okay. Okay, Kausa, is it okay?
Yeah, Prof, thank you. It's very clear.
Okay. Okay, any other questions? Okay, so if there are no questions, then that ends our presentation. Okay, I will email you Kamarina's presentation later, and you will have to email back to me the answer. Okay, we still have 1 minute. Okay, so for that, let me share with you regarding the exams. Okay. Okay. Okay, for the exams, for the mid-semester exam, it will be next week. Okay, 9 to 11 a.m. That is meaning that it is during class, that this will be on Friday. Okay, and then questions, I will upload it on UKM Folio at 9 a.m. so you can access, and straight away you can start answering it. Okay. So lecturers, meaning me and also Dr. Shu, will be present on Google Meet. Okay, same link as today. Okay, our class link. Okay, if you have any questions, okay, if you want to ask straight away, or if you want to, you do not have to be present in the Google Meet. Okay, but if you want to ask directly, then you can join the link and then ask, or you can type it in the chat, or you can also WhatsApp us. Okay. Okay. Examination is by open book, meaning that you can look at your notes, you can look at any other sources, the internet, etc. But you are not allowed to discuss. Okay. Of course, there is no way for me to really monitor that. Okay, but we leave it to your honesty. Okay. Okay, so answer all questions within the allotted time. Okay, and then save Okay, there will be 2 parts for 2 hours. Okay, but I think you can finish it within 1 or 1.5 hours. Okay, so there is ample time for you to answer all the questions. Okay, so part A will be Dr. Shuhada's part. Okay, so separate your answers. Okay, save part A answer file as STKM6122 part A, and then P whatever your matric number is. Okay, please in PDF format, and then email it to Shuhada, to Dr. Shu. Okay, name the delivery email as STKM6122 part A and your matric number. Okay, similarly for me, okay, email it to me. Okay, and I will be giving you an additional 30 minutes. Okay, to submit your answers, after which it will not be accepted. Okay, you can answer in Malay or in English, it doesn't matter. You can also answer it using handwriting or using computer. Okay, as long as it is legible. Okay, any questions? Okay, so if there are no questions, then that's it for today. I wish you all the best for your exam next week. Okay, and for Xiu Yan, Happy New Year. Happy Chinese New Year.
Thank you,
Okay.
Prof.
Okay, welcome. Okay, so that's it. Okay, thank you, everybody.
Thank you, Prof.
Thank you, Prof.
Thank you.
Prof.
Thank you, Prof.